Thank you. I want to thank the organizer. The lineup is very impressive. I'm really looking forward to all of the talks. So Seraphim laid out the foundation for a lot of what I'm going to talk about. So I'm going to ignore basically the analysis. I'm going to focus much more on the interpretation of single-cell data, and specifically focus on time series single-cell data. But before I start discussing networks and regulation, I want to talk about two issues related to the analysis of time series data that I've been thinking about and trying to look at for a lot of time, and at least a decade. And we have some results recently. I'm hoping that other people will generate even more results in that direction. So time series data, the thing that we started with, and most of the people initially thought about, was gene expression data, primarily microarray, now a lot of RNA-seq. But over the last, I would say, 2, 3, maybe 4 years, a lot of other types of data are measured over time, a lot of it because of the sequencing, so microRNAs obviously, but also a lot of epigenetic data is now measured over time, histone modification, you can do DNAs and so forth. Protein levels have been looked at over time, but more so now. And also single-cell analysis has been looking at development as a time series. And there are two basic issues in the analysis of time series data that I think we have not addressed, at least not as much as we should have. And a few years ago, I was asked to write a review on using time series gene expression data. And the editors really wanted me to focus on the high level, what I call the high level analysis, so differentially expressed genes, clustering, modeling, and so forth. And I really wanted to discuss something that I thought that people don't pay enough attention, in the sense that when you work with a group of collaborators on the analysis of time series, they usually come to you after they did the experiments, and then they tell you, let's do the clustering. I was really worried that we should be involved much earlier. So the thing I wanted to discuss was how to design these experiments. And we went back and forth, and eventually we agreed that it wouldn't be in the paper, but it would be a separate box, which is where it ended up. So this is the sampling rate. But in that paper, we mentioned two issues that are related to time series data. The first is repeats, and the second is the sampling rate. If you look at time series data in public databases, you see that most of them, on the order of 70%, and don't contain repeats. And the reason is clear, right? If you do time series, you don't have enough money to also do a second and third repeat for each of the time points. If you have two, very, very little have more than 2 repeats per time point. Now, over the years, I've been contacted by a number of researchers that were telling me, look, I submitted this paper, and the reviewer came back to me, and because I wrote some reviews before on time series gene expression, the reviewer came back to me and told me that, it's unacceptable. I did time series, and I didn't do any repeats. I don't want to spend another 4 months and tens of thousands of dollars on this. What can I tell him? And the best answer I could give him is, look at this paper and this paper and this paper. They also had time series with no repeats, and you can say that's pretty common. But there was no, or at least I didn't see, there was no theoretical analysis that really told us that, maybe we shouldn't, or at least if there are constraints in terms of budget, which we always have, maybe we don't have to do repeats. So that's one issue. The second thing is the sampling rate. Now, if you read a time series paper, Genomics time series paper, again, this is more anecdotal than the previous statement I made, but 50% of them don't even tell you why they selected these points. They said we sampled week 1, week 3, week 4, and maybe some people in the domain know, but most people just. The other 50% do explain why they selected it, and almost in all cases, the explanation is based on phenotypes. So we know that at day 4, this marker is expressed, and at day 20, the lung is developed. So that's why we do these experiments. Now, this is important, but obviously, if you want to find out why this marker is expressed, maybe you want to sample a bit earlier to see the networks that led to that. And there was no standard or no protocol to tell you what is the best time points to sample. Now, again, if you just do gene expression, maybe that's not so bad. But now you do gene expression and epigenetic and proteomics and microRNAs, and you want all of them to be sampled at the same points if you want to build models. So maybe there's a better, or maybe we should come up with better ways to systematically decide when to sample. So we looked at these two issues recently, and again, these are things that I've thought for a number of years now. So the first issue is repeats, and we can now show theoretically that it's better to sample at a denser rate and ignore the repeat. Let's say you have a limited budget. For example, you have a budget to do 20 experiments, let's say in 10 hours. It's better to sample at a denser rate, so every half an hour for 10 hours, rather than doing every 1 hour with 2 repeats in the ability to reconstruct the underlying expression trajectories. And again, this only works for expression trajectories, and this only works for gene expression where you are sampling a lot of different signals at once. It doesn't work for individual signal. Okay, but this works. And when I say theoretically, I mean that you can always empirically go to papers and see what happens when you remove some samples and so forth. But this doesn't cover the entire universe. We actually can show now that for a specific type of function, piecewise linear function with relatively small number of transitions. So if you only assume that the gene can go up and down maybe, so not too many, which is actually, it's a constrained space, but it's pretty characteristic of characterizing the entire space of time series data. For those, if you have a reasonable error rate, which is common, dense does better than repeat in terms of error. So this is dense. Of course, once the error increases, then they both do more or less the same. But for a reasonable error rate, dense is much better, and you can look at a lot of things. So this is one result. Again, very more theoretical, maybe justification. But the other result is a bit more practical, and that's how to design these experiments in terms of how do you choose the sampling rates. And for some reason, we had this idea where you can basically do a very dense sampling. Basically, for example, if you look at 42.6, we are looking at lung development, and I'll talk about it in this presentation. If you if you look at 6 weeks of lung development, you can sample very densely, let's say every half a day, for relatively small number of genes. You don't want to do 80 or 100 RNA-seq experiments, maybe that's too expensive, but you're willing to use some bead microarrays to do a very detailed sample of a small set of genes. And it turns out that with this subset of genes, relatively small, 150, 200 genes, which can be based on some prior knowledge of what you know is involved, but it can also include a lot of random genes, you can actually determine a very good sampling rate for the rest of the population. And then the good news, at least from our point of view, is that the decisions you are making based on the gene apply for the microRNA as well and the proteomics. So basically, you learn the sampling rate based on the genes, but then you apply them to all other data sets, and it works reasonably well. Yes, Bill?
Just a clarifying question. I don't understand the vertical axis is a mean squared error on quantification of gene products and genes. Is that right? And
no. So what you do is you select a subset. So in this case, we started with 40 points. You select a subset of the points, you reconstruct the entire expression profile based on that, and then you compute the error on the ones you didn't use. So this is, you can think of it as held out data. Okay, so in all of them, the held out data is, or basically the error is on the held out data, the points you are not sampling, basically.
I guess I'm confused about if you have different sampling rates, one is sampled more densely than the other. It seems like how
you can see that it goes down, right? The more points you have, it usually goes down.
How you compute the y-axis on points that are sampled at different times.
The y-axis is per a set. So basically, the setting we are thinking of is you have a limited budget. You can do 10 experiments, and you have 40 days. Where would you put those with your budget, right? So you have to select. Once you selected the 10 points, you reconstruct the entire curve from that for the entire 40 days. But now we did sample the other 30 points, and you can compute how much error, what's the reconstruction error you get. So this is like an held out set, okay? So of course, to establish the method, we actually did oversample everything, but this is the error that we're computing. Okay, so now let's go back to networks and now assume that we know how to sample and everything is optimal and we get that data. Okay, so I want to talk about reconstructing networks in single cell analysis. So we've been looking at methods for integrating various types of genomics data, specifically time series data with static data. And again, in many cases, the time series data has been gene expression, but you can also, we've also used over the years microRNAs, ChIP-seq time series data, histone modification time series data. And the static data is primarily ChIP-seq static information, motif information, sometimes protein interaction information. Now, in order to integrate these two, you want to have a model that retains the dynamic aspects of the data, so you can actually tell what happens at which time point, but can still use the static data to infer events or regulatory events. And for this, we're using a method called input-output hidden Markov model. which is a standard hidden Markov model, or includes a standard hidden Markov model where states represent, basically in this case, gene expression. You have emissions for the genes, but you also have this input layer, which is static. And you encode all the static informations, for example, the transcription factor, binding preferences, or a microRNA regulation in this static data. And what happens is that the static data does not directly impact the expression, but it does direct the transition probability. So if you have a gene that is expressed at some level, the probability that it will go up in the next time point is a function of which transcription factors regulate it. Now, once you know that it goes up, the actual expression is coming from some distribution. So basically, the likelihood of this HMM is very similar to regular HMM, but the transition probability is not just a function of the previous state, like or the transition of the HMM, but also a function of the static data. Now, again, this was developed a few years ago, and over the years we applied it to a lot of different, and others, not just us, to a lot of different types of networks, and it worked reasonably well. But now, when you deal with time series single-cell data, you run into some new problems, which is nice. So one of the major, so Sarafim introduced single-cell data. Obviously, when you do these experiments, you have a lot of problems relating to how do you infer the gene expression levels and so forth. Let's assume that we know how to do that. But one of the major, but if you do the time series analysis, you run into the following problem. So usually the experiment starts with a set of cells that are undifferentiated, maybe stem cells, and it ends in what researchers at least think is a differentiated set of cells where they can be or they can be sorted by some marker. And that was a great comment before about the fact that even though we know the markers, we may actually not know all of the markers. I think it was your comment. That's a great comment. Let's get to it a bit later. But for now, let's assume that we know how to sort the cells at the end. So we have these cells at the end. And then you measure a number of intermediate time points. Let's look at one time point. And you want to create trajectories that link the stem cells to these final or differentiated cells. And here's the challenge. Usually you know the starting point, you know the end point, but you don't really know what should be used to link the intermediate points. And once you take a cell out and you profile it, that's it. You can't see where it differentiated to. So it's easy to deal with the first time point, easy to deal with the last, but the intermediate time points, which are the more interesting, are very hard to associate with specific trajectories. And if you want to learn now models of a cell fate decision or regulatory networks that impact cellular differentiation, we need to be able to create the trajectory or determine cell assignments to this trajectory. So that's a major problem that people have been looking or noticed over the last year, I would say. Right, over the last year. And an additional problem is the fact that the time in which you measured the cell does not necessarily correlate with the function of the cell. What do I mean by that? And this actually has been shown very nicely experimentally. So it could be that even though we are measuring cells at maybe 3 different time points, some cells at the last time point, in this case, let's think of lung development, so we're measuring at day 14, day 16, day 18. Some cells at day 18 are really still undifferentiated, and they belong in some sense to the intermediate cells, but they are mixed in with the differentiated cells. And that's true definitely for in vivo development, and it's even more true for in vitro studies. For example, people have looked at blood or myoblast differentiation. So it turns out that even if you do a time series, the one thing is how do you connect these cells along the trajectories, and second, do you really trust the time of the experiment to do any of these assignments? So, for example, are you always assigning the second cells in the second time point to intermediate or maybe to that? And these are 2 issues that people have identified or looked at. And there's 2 major approaches to dealing with this. And these came, of course, in the last year. These are pretty new studies. The first set of approaches, and again, I don't want to sound criticizing. They looked at slightly different types of data sets, but basically they completely ignore the temporal aspect. Basically, if you sample cells at different time points, they ignore the time, and they created what they call pseudo-time. So basically, they order the cells based on just on gene expression alone. And some of these allowed for branching, some didn't allow for branching, and that's a separate issue. But they looked at that. they also, I should say, used, and again, I like the point that Sarfi made, these methods use deterministic approaches for computing the similarity. They looked at dimensionality reduction using minimum spanning tree, so there are some issues related to stochasticity, but that's a separate issue. The second approach was completely ignore the expression and just believe the time. So this is the time. Again, that doesn't solve the trajectory problem, but at least it allows you to order the... And both of these may be problematic, as I think I tried to explain. And of course, all of the methods so far have only relied on the gene expression. They didn't integrate other types of data, for example, transcription interaction. So we asked whether we can improve some of this by coming up with a new model that takes into account both the time and the expression and allows for stochasticity in both measurements. So one approach we are looking at is an HMM, different from the previous one. I'll go back to that later. But this is a standard HMM, except for one thing. The emission and transition probabilities are linked over time in the sense that they are autoregressive or Kalman filter generated from the previous time point. So each of these states is basically a collection of cells, or represents a collection of cells at a specific time point. And so they have an emission probability related to the gene expression and to the time. So cells from the first time point usually have a time point from the first time point, but they can also have time points from the second time point because of this asynchrony. And also they have these gene expression trajectories. Okay, so this is standard HMM in terms of emission, but the transition probabilities are slightly different than regular HMM. First of all, we fix the model, or we look at slightly a number of different models, and we have transitions, but sometimes we only allow transition from a previous state. But the mean values for the expression in time are evolving from the previous state. In other words, you have a Kalman filter model for the expression, or autoregressive model for the expression of the next time step conditioned on the expression in the previous time step. Okay, so this mean expression, of course, is a function of the cells assigned, but it's also a function of the previous state. In some sense, you assume that gene expression and time are highly correlated between consecutive states. Now, again, if you do a time series analysis in bulk cells, that's clear, right? Because the next time point is but in this case, we don't just need to associate the expression, we also need to assign cells to these states. So if you don't require it, you can get very coherent states, but they can be all over the place in terms of their relationship. So you need to somehow constrain these transition probabilities. And I'm not going to go into inference and learning. Learning in this case involves a nested EM, because you have, in addition to the regular EM, you need another forward and backward to estimate the expression based on the autoregressive model, and then you do the regular EM learning for the assignment and for learning the parameters. We'll skip that, but I'm going to talk about applying this to some data, and specifically the data I'm looking at, again, lung development data. That's something we've been working with collaborators for a few years now. And the original the experiment was done 2 years ago, and they looked at 3 different time points, 14 days, 16 days, and 18 days. And again, measured a number of different cells, of course, but the focus was on the last time point. So basically, they had markers for these last time point. Of the cells that were assigned to the last time point, a lot of them, roughly 80%, displayed known markers. 20% did not display any of the known markers. And so these were assigned by the researchers to a new cell type, which is called bipotential progenitor. Basically, this is a new cell type that was discovered by these experiments because it didn't match any of the known cell types. Okay, so let's see what happens when you try to analyze this data with different methods. So this is the first method. This is the monocole that I mentioned that doesn't use time at all. So it just takes the expression and tries to figure out the branching process based on that. And it doesn't work well. And again, not surprisingly, it doesn't use the expression. Here's what you see. This is the first time point, the purple one. Then you have a branch for the second time point and the third time point. So you don't go really from the intermediate. They just clustered somewhere on their own. There's no real trajectories. And the third time point is also, so each color here is a different type of cell. You can see they are broken up almost randomly in terms of, one is the majority of the cells are here, but then you branch into a different, again, this is biologically doesn't make sense. That's actually not true biologically. And one of the major reasons why this fails is because there's no, I mean, just based on the gene expression, you can't probably infer, or at least not with this method. This is the results that we get using this HMM model. And in this case, I didn't mention, but of course, the HMM, the way I presented it, is applied to a specific model. So what we do is, and you can do different search methods, but right now what we do is we look at a number of different possible models, and the nice thing is that you are looking at a very small number of time points, so the space of possible models is pretty small, and you compute a BIC score, and then you can compare different models. And in this case, the model that was selected by HMM is this model, which is basically starting with one cell type, then 2, and then 4. We actually tried, I don't have it here, we also tried something that splits to 5, but it scored actually much lower than the 4 model. So in other words, the model that we have claims that you have this branching process. So, and again, I'm not showing you the cell assignment, I'm just showing the branching process. But if you look at the cell assignment, you see that each of the 4 different cell types that were known is associated with a terminal branch. And again, this was unsupervised, so the fact that it's at least to some extent a biological validation that the model makes reasonable findings. So you can see each one of these is associated with the terminal branch. There's only one set of cells that is not associated with a terminal state, and that's these new type of cells. In fact, not only are they not associated uniquely with a specific type of cells, a lot of them are associated with the earlier time points. What this basically model claims now, and this is a biological finding, that these bipotential progenitors are not really a new type of cells. These are just earlier cells that have not fully differentiated. And in some sense, they actually should be associated with the previous time point. And if you look at the biology behind this, you see that this is actually something that you can explain, or this agrees with some other models where you have a number of cells that can differentiate later to a different type of cell, and not, or in this case, 81 or 82, the 2 primary epithelial cells. And they don't have to actually be fully differentiated at the last time point. So again, if you just use time, you get one observation, which is we have a new type of cell. If you just use expression, it's very hard to reconstruct. But if you combine both of them, you get models that at least make sense. And there are other things you can do with it. Let's skip to the next. OK, so once we have this, but let's go back to the models. Once we have this, we can actually now learn a regulatory model. How do we do this? So again, we start with the set of cells. And let's say we have some assignments of the cells to different states based on the initial model that we learned. For example, based on this HMM. So now once you have this, you see that these terminal states are actually using the same intermediate cells. So both of them are, the progenitors of these two are these cells, and the progenitors of these are these cells. So now, basically, you have a new trajectory of cells, or a new time series, basically, that you can use to learn, and with any of the methods you like to learn regulatory networks from time series data. Again, we like to use our method, but anybody can use any method. There's a few other things you want to do. So this is, for example, one of them. You can derive regulatory networks based on these DREM models for this specific type of cells by just taking the cells that are assigned and treating it like a regular bulk expression data set now. Because now we assume that the cells are more homogeneous in terms of the different state they are associated with. Seraphim mentioned it. You have to be a bit careful when you deal with single cell data. And one of the major problems is the dropout issue. So dropout means that genes that are expressed at low levels can be completely missed. So it's like almost a binary signal because of amplification and other things. If you have a probabilistic model that doesn't take that into account, you can very negatively affect the association of cells with state. Because if you have some probability and it's 0, basically, it can be very bad. So this is something you need to deal with, and there are ways to, in this case specifically, we're looking at mixture probabilities for emission, but these are not necessarily very complicated. Right, so we have an iterative assignment of cells like we did before. Another thing we are very interested in is identifying not just the models, but what are the transcription factors that differentiate the different models. Why is that important? Because if you look at cell find determination, you want to explain why some cells become this type and other cells become this type. For that, you want to identify what are the regulators or the key regulators that differentiate the two. And if you know that, you can actually treat the cells or maybe knock out these and see what is the outcome that you get once you do these perturbation experiments. And if you're right, maybe you can actually you can actually show that these are the ones that lead. And we have some way of determining significance for these transcription factors based on how well they explain or how well genes that are regulated by them are differentiated between the two models. Okay, so this is something we apply to the same data set that I mentioned, this lung data set. And you learn, in this case, 4 different models, one for 81 cells. And this, by the way, this method allows you to reassign cells. So initially, we assign cells just based on expression. In this method, we use expression plus other information, in this case, transcription factors. So you may actually get slightly different results. We have allowed, so the method is allowed to reassign cells to states. And it turns out, but in general, it finds more or less the same result. We have one terminal model for 81 cells, another one for 81 because one of them was very small. But the important thing is that the BP cells are divided between the 82 model and 81 model. So the same conclusion. You can also find, as I mentioned, transcription factors that distinguish the different cell types. Many of these transcription factors are known, and you can explain why they are high in one type. You can actually explain why they are high in one type, or they are actually known to be involved in one type of cell but not in the other, and vice versa. So in that case, that's some validation that the method does identify not just models but also transcription factors that drive that. And last thing, we looked at some experimental validation. And again, there's different things you can do. It's very hard to do this in vivo studies, and this is in vivo data in terms of perturbation. So we took a much easier approach, I would say, in some sense. And we looked at some transcription factors that came up in the models, in this case in the 81 model and the Foss-June family, but did not come up when you do the bulk analysis. So if you take all the cells, put them together, you don't see a unique pattern for the Foss and June transcription factors for this specific system. Only when you separate it out computationally to the different models, they start to appear as key transcription factors in one model but not in the others. So basically what this is a prediction based on a specific type of model, but not as bulk. And fortunately, we were working with collaborators that had an IPS system set up to express specific types of lung cells, in this case, human. So the experiments were done in mouse. The collaborators had human mouse cells. And it turned out that these specific transcription factors that we identified in the 81 cells indeed are differentially expressed, and their targets. So these are some of the transcription factors, and some of their targets are differentially expressed in this IPS system as part of development. So again, it's very hard to find them when you work on bulk cell, but when you work on single cell, it becomes easier. I think the ultimate experiment, which is very hard to do, but we're hoping to do, is to do some knockout or perturbation and see that we really get much fewer types of cells from the ones that the transcription factor is believed to regulate. Okay, so that's it. I think single cell data, on the one hand, it's very similar to issues we have been discussing for almost 20 years now. How do we find differentially expressed genes? How do we normalize and so forth? But how do we identify time series, or how do we model time series data? On the one hand, these seems to be very similar questions, but on the other hand, they have, I think, a new dimension or some new challenges that we didn't have with the previous type of data. And I think they provide, or they can enable us to answer a lot of questions that are very hard to answer when using bulk data. So I think it's an exciting time. Actually, it has been an exciting time for the last 15 years in this area, but it remains an exciting time. So with that, I want to thank a lot of my students and collaborators who have worked in DRAM over the years on the single cell data. We are collaborating with a group at BU. And that's it. Thank you.
If I'm having trouble getting my head around your common extension to the HMM, can you just give us an intuition about the generative process that underlies this model?
Right. So the generative process is that you have basically the generative process is single cell differentiation, right? So you have a set of cells, a set of stem cells, right? And due to either external cues or some internal cues, they start to differentiate into more than one type of cells. Now, the idea is that even though they differentiate, most of the genes are not affected by this cue. A few of the genes are affected. Most of the genes are not affected. So if you have cells that are coming from again, depends on the sampling, but if you have cells that are very close in time, you expect the gene expression pattern to be pretty similar to each other. And also, you would like, or you would expect their time to be more or less similar. Now, there could be things that, you measure something very late, which just took a long time. So this is the generative process. Generative process is the gene expression changes relatively slowly. It does change, but it changes relatively slowly. And consecutive time points should be highly correlated, basically. That's the
So I was wondering whether the assumption about expression changes being relatively slow is not affecting your difficulty with June phosphate, because those are immediately early genes that pop up really, really quickly, and they cool down also very, very quickly.
Yeah. So
is that particular family?
Great question and great comment. So gene and force are transcription factors. Transcription factor, especially the key transcription factors, are. First, some of them are activated early, and some are actually even post-transcriptionally activated, right? So you don't even see them activated, and they are there. There's been two lines of thought in the big community about how to model transcription factors in studies. One line says, let's look at their expression pattern and see if they're active, then we say, or if they're changing, we can say they're active. We take a different approach. We only infer that the transcription factor is active based on its targets. So it doesn't matter how long, at least from our model, how long June and force are up. As long as their targets are up for enough time, we can identify them. But yeah, it's a good point.
So if I understand correctly, you are getting different networks for different cell types?
Yeah. Well, we are getting different. The overall networks are different. They share the initial time. They can share substructures, but the overall networks are different. Yeah.
Because I guess some of these cell types emerge as part of this developmental process.
All of them emerge. Yeah.
Still you have a network, separate network starting at time point 0.
So again, the question is, what do you call separate network? So we have, let's say we have 4 different, or in this case we have. I don't know if you can see here. You can see that these two look pretty similar initially, right? And these two, at least these two look pretty similar initially, right? The reason is that we believe that the cells that lead to, but at the end they look slightly different. Maybe this has this big jump here, which this doesn't have. So what happens is that, they share components earlier on, but these components do not necessarily have to be shared in the entire network. So you can have subsets that are similar and subsets that are not similar. Because of the way the HMM is computed, later time points can have impact on some earlier time points as well. So you can have but in general, again, if this was longer, they definitely share a lot of the initial structure. And that's by design. So yeah, you're right. It's the same process, basically.
Maybe we should now move on to the next talk. Thanks again.